Perfect. So, hi everyone. My name is Dexiong Chen. I'm from the Machine Learning and Systems Biology Department of Max Planck Institute of Biochemistry. It's my pleasure to present our work entitled Biomarker Identification by Interpretable Maximum Mean Discrepancy. This is a joint work with Michael Admer, Sarah Bruning, and Karsten Bogwat. And this work was done during my postdoc at ETH Zurich. So in many biological studies, we aim to find differences between two groups of samples, such as treated and control groups. And this task often involves determining if the groups are statistically different, which is usually called two-sample hypothesis testing in statistics, as well as identifying the features that best distinguish them, which is often called feature selection in statistical learning. However, existing methods often tackle these tasks separately, leading to challenges in interpretation and efficiency. So in this work, we attempted to offer a solution by integrating both steps into a single and powerful framework. So as a background, two-sample testing assesses whether two groups of samples are statistically different, while feature selection aims to pinpoint the specific features that contribute most to these differences. However, there are limitations with current methods to deal with both problems simultaneously. For instance, while univariate tests can handle two tasks simultaneously, they can only analyze one feature at a time and often overlook complex interactions between features. And thus, they can lead to false positives due to multiple testing. On the other hand, multivariate tests may offer greater statistical power, but often lack interpretability, making it difficult to understand the biological significance of each feature individually. So, in particular, kernel-based maximum mean discrepancy tests are powerful multivariate tests, but they cannot perform feature selection. So, in this work, we provide a solution to this problem, termed spin-opt MMD. This method is designed to overcome the limitations of existing methods. It is a sparse, interpretable, and optimized MMD-based two-sample testing. So I'd like to first emphasize here several fruitful features of our method. First, our method seamlessly integrates multivariate two-sample testing and feature selection, providing a comprehensive view of the data. Second, by directly optimizing feature weights, it offers interpretable results and highlighting the most important features driving the differences between groups. Moreover, our proposed spin-off MMD achieves superior test power compared to traditional MMD approaches, making it more sensitive in detecting subtle differences. And finally, its versatility allows it to be applied to a wide range of data types and modalities, including images, gene expression data, and text data that I will show later in this talk. So the maximum mean discrepancy is a statistical measure that quantifies the difference between two probability distributions, which is defined formally in this equation 1. It operates in a reproducing kernel Hilbert space, and in particular when using a characteristic kernels like the RBF kernel, MMD becomes symmetric on distributions, meaning that it satisfies the properties of a distance measure. And there are two commonly used estimators for calculating MMD in practice. The first one is MMD linear with a linear time complexity, and another one is called MMD quad. While MMD quad has a higher quadratic time complexity, it is statistically more powerful and particularly useful in scenarios with moderate sample sizes. So its expression is given in this bottom equation. So here, now I will illustrate how the maximum mean discrepancy is used for two-sample testing. So the goal of two-sample testing is to determine if two sets of samples drawn from potentially different distributions are statistically distinct. And MMD provides a way to quantify this difference by checking whether the empiric MMD statistic is too large. So here, too large is quantified as it falls outside the 1 minus alpha quantile of the new distribution for chosen significance level, alpha. And then we can reject the new hypothesis and conclude that the two samples are significantly different. In practice, the new distribution can be estimated using bootstrapping a resampling technique. And this figure shows an example of the estimated new distribution. So if your observed MMD fall in the extreme tail of this distribution, then you are likely to reject the new hypothesis. However, the standard MMD test, as illustrated here, considers all features equally when distinguishing between the two groups. So it does not have a mechanism to select or prioritize specific features that may be more relevant for this differentiating two groups. And this limitation is addressed by our proposed method, which we will introduce in the next slide. So our proposed spin-opt MMD enhances the RBF kernel used in MMD by introducing feature weights. And these weights are optimized on a training set using an objective function that combines the MMD quad estimator with an L1 regularization term. So in this equation 2, the first term measures the test power of the MMD quad estimator, and the L1 regularization term encourages sparsity, meaning that only the most important features receive non-zero weights. And this optimization process not only maximizes the test power of the MMD test, but also identifies the features that are most relevant in distinguishing between the two groups. And this problem can be solved using a stochastic gradient descent method in practice. So the previous process offers us a good subset of features to start with. However, the selected subset can be often noisy on small datasets. And now to refine the selected subset, we propose a simple greedy heuristic approach to identify the minimum set of features that maximize the change in MMD. And the selection algorithm starts with the empty set S and iteratively removes features and evaluates their impact on the MMD statistic. If removing a feature with a positive feature weight causes the most significant drop in MMD, then this feature is considered important and added to the selected feature set S. And this process continues until the MMD falls below a predefined significance threshold, indicating that the remaining features are not significantly contributing to the difference between the groups. And to evaluate the performance of our algorithm, we used a diverse set of tasks. We started with synthetic toy datasets, including Gaussian mixtures and blobs, to assess our method's behavior in controlled scenario, and you can find our results in the paper. In this talk, we will focus on the benchmark and biomedical datasets, which are more interesting. So the benchmark datasets include NeurIPS papers and Karolinska phases, which are commonly used in the machine learning community. And the real-world biomedical datasets contain ADNI, which is a dataset for Alzheimer's disease neuroimaging initiative data, and TCGA-LGG, which includes both gene expression data and imaging data from the Cancer Genome Atlas low-grade glioma, and PBMC, which includes gene expression data from peripheral blood mononuclear cells dataset, and DRAMAS, which is a database of resistance information on antimicrobials and Malitov mass spectra data. And for comparison, we use the three baseline methods, including the classical MMD linear with the linear time complexity and MMD quad with the quadratic time complexity. And also a recently published interpretable two-sample test proposed by Jit Cretan and colleagues in 2016. We evaluated the performance of our proposed spin-opt MMD and the baselines based on several criteria. First, we assessed their statistical test power, which is their ability to correctly detect a true difference between groups, and also the type 1 error rate, which is the probability of falsely rejecting the null hypothesis when it is true. We also evaluated the quality of the selected features by examining their performance in the corresponding binary classification tasks. And finally, we qualitatively assessed the identified features to see if they aligned with known biological knowledge or provided any new insights. So we first showed the performance of our proposed spin-out MMD on the benchmark datasets. The Karolinska faces datasets contain two groups of face images with different emotions, and the NeurIPS paper dataset is to distinguish categories of publications at NeurIPS, a top-tier machinery conference. As you can see in these tables, our proposed spin-opt MMD consistently demonstrated superior performance compared to the baseline methods. It achieved a higher test power, meaning that it was more likely to correctly identify true differences between groups. While achieving higher test powers, the type 1 errors of our proposed method are similarly low to the baseline methods. And notably, our method is also able to find discriminating words when distinguish these categories in the NeurIPS paper dataset. So if you are interested, you can find the top discriminating words in our paper. And in our experiments on biomedical data, our proposed spin-opt MMD consistently outperformed the baseline methods in terms of test power, indicating its superior ability to detect true differences between two groups of biomedical data. And in this table, the test Q means the test power of the MMD quad estimator used in different methods. And the test L means the test power of the MMD linear estimator used with different models. And notably, the figure on the right compares the classical MMD and our proposed spin-opt MMD in terms of type 1 errors and statistical test power across different biomedical datasets. And you can see that our proposed spin-opt MMD consistently outperforms classical MMD in terms of both metrics across all datasets. And now to assess the quality of the features selected by different methods, we evaluated their performance in the corresponding binary classification task as a surrogate way, since we don't know the true discriminating features in these real-world datasets. And for this, we used the logistic regression models to predict binary outcomes in 5 different biomedical datasets, using either the entire features or the selected features by different methods. And this table shows the classification performance measured by ROC AUC for different feature selection methods, including our proposed spin-opt with the MMD quad estimator and the shape analysis method, and also a few univariate feature selection methods. And you can see that our proposed spin-opt MMD consistently achieved better performance than using all features. It also outperforms the shape analysis method and univariate feature selection methods, showing its ability to select informative features that are relevant for discriminating the two groups. And we also studied the overlap of the features selected by different methods. So the overlap analysis in this bottom figure reviews that our proposed spin-opt MMD may identify a distinct set of features in some datasets compared to univariate feature selection and shape analysis method. As you can see on the left of this figure, all methods select a very different set of features. This suggests that our proposed method captures unique information that may be missed by other methods. So finally, we also provide a case study in the context of imaging data from Alzheimer's disease. And this figure here demonstrates the ability of our proposed spin-opt MMD to pinpoint specific regions in MRI scans that are significantly different between healthy and diseased individuals. And in this case, our spin-opt MMD successfully identified regions in the hippocampus, a brain area known to be affected by Alzheimer's disease, further validating its potential for biomarker discovery. And the selected features are further validated by observing the improved classification accuracy when only using the selected pixels with a convolutional neural network. So you can see that using the selected features to train the convolutional neural network improves the classification accuracy from 81% to 85%. And finally, to conclude, we propose the spin-opt MMD, which is a powerful and interpretable method for simultaneous multivariate two-sample testing and feature selection. Our proposed method outperforms existing methods in terms of statistic test power and feature selection accuracy. And our proposed approach is applicable to a wide range of data modalities in several biomedical applications. And our approach potentially opens new avenues for biomarker discovery and data interpretation. So thank you for your attention, and I'm happy to take any questions.
Sorry,
Okay.
you should be able to hear us now, right?
Okay.
Can you hear us?
Yes,
There
I
we
can
go.
hear you now.
Okay.
Yeah, sorry, I'll repeat the question. So have you looked at the correlation between the features that your method selected versus some of these other methods, for example, in your TCGA analysis?
Yes, so we look at the the overlap of the selected features between different methods, but we found that so maybe I can share that slide again. So for instance, on this TCGA LGG gene expression data, which is a relatively small data set, and we found that actually the selected features could be quite distinct. And there's no way to validate the selected features. So basically, in some cases, we found that there's some big overlap, as you can see on the right figure, which is on the radiomic data set from TCG LGG. But in some cases, different methods might select a very different set of features.
Thank you. I was wondering, for example, often in this type of data sets, you have multiple features, multiple genes that are highly correlated. And when you have selection methods like what you described versus some of the other ones, there's sort of theoretical and practical issues in how one might be able to best select features. So I was just wondering if you have looked into those. So you have genes that are highly correlated, for example.
So we didn't study this empirically, but at least when comparing to univariate feature selection methods, our method has an advantage because it is a multivariate feature selection method. So at least it looks at multiple features simultaneously, which might be might be less sensitive to like correlations between multiple feature selection. And also it usually select a pretty sparse set of features because of the L1 regularization that we imposed to the method. So I think at least if you only compare our method to a univariate feature selection, our method has obvious advantages.
Thank you. Any other questions? Up here.
Thank you for the talk. I have one question basically. So I was wondering if this method you proposed count for example, the batch effect in your input data, or like a sample to sample heterogeneity.
Yeah. So in our paper we used already like pre-processed data, so it does not really take into account batch effect.
Okay.
But you could like combine our method with any like pre-processing or normalization methods.
Yeah, so the question I ask is basically because we have a very practical issue. For example, in the clinical trial RNA-seq data, like a patient-to-patient heterogeneity, like a very large. Sometimes it can just like if you do the differential expression analysis or other comparison between like pre and post-treatment, if you average those gene expression, you basically cannot find any like differential expressed genes. I was wondering, I may have missed it, but I was wondering if the algorithm you proposed could potentially help solve that issue.
I think totally depends on the size of your data and also like the dimension. And as you can see from our paper, so our method successfully selected the hippocampus area for this Alzheimer's disease data. So which shows that, and actually our method was applied to the raw pixels of the image data. So basically it could be quite high dimensional. So if your data is of high dimension and with moderate sample size, probably you could try our method. But it really depends on like the specific data types and also like data set size and dimensions.
All right, thank you.
Thank you.